Alright, welcome to the Unit 5 Notes video. As always, you should have your notes printed off as PDF slides. You can do that from my website. You should also have a pen or pencil handy to write some things down that I will tell you to write down as we go. And you might also want to get out a highlighter. I'm going to give you some things that you can highlight along the way that'll just kind of help organize things and stress and focus things you want to spend a little bit more time on for when you do get around to studying for the next test. So here we are, Unit 5. We're going to transition pretty nicely from our last unit talking about transport. You can see how we're going to discuss how transport affects cell size. One of the things I'm going to ask you to highlight first is objective number 3 here. It's a calculation. It's something you're going to have to do. We're going to do a lab on it, so it's kind of a big deal for this next unit. Not that it's any more questions or anything on the test, but it does consist of a big part of this next unit. Calculating surface area to volume ratio. And actually there'll be a separate video for that. So let's move on to the next set of objectives. Here you can also highlight number 7 and number 11. The phases of mitosis are going to be a big section of this unit as well, and you'll also have a separate video for that. Objective 11, where it's talking about DNA replication. This is basically the third chunk of this test. So it's going to be talking about cell size and surface area to volume ratios, chunk number 1. The second chunk will be the 4 phases of mitosis. And then the third chunk will be DNA replication, which is part of just the overall cell cycle. Which is cell cycle and DNA replication, you can see the main focus of this unit. So let's move on to the first slide. You can see right away the segue from the last unit, how we were just talking about things like diffusion and osmosis, transport through a membrane into and out of a cell. So we're going to start with a question here, and this takes us back to our last unit. The process that cells rely on for transport of lots of things, at least passive transport, is diffusion. Remember, we talked about diffusion in the last unit quite a bit. So cells rely on diffusion for transport of quite a bit of materials, gases, and other things. And how this relates to our new unit, Unit 5, is talking about cell size, cell cycle, cell division. And your next fill in the blank here, as cells get bigger, transport, at least transport by diffusion especially, just becomes too difficult. Right? It's too hard to transport things by diffusion if the cell is too big. A good example to think of that is maybe thinking about Febreze, and I talked about Febreze in the last unit, and that getting to your nose by diffusion. Those molecules are just being pushed through the air by a concentration gradient, high concentration to low concentration. You know, let's say the Febreze company, which is based out of who knows where, USA, let's say California. You know, you live on the opposite side of the country, and they came out with a new scent. Well, they're not just going to start spraying it into the air and hopefully it gets to your nose. You know, that's diffusion. That's impossible. So they're going to transport it, you know, on a truck to the store. You know, all these other things that are requiring energy. Diffusion would be just impossible, and that's kind of the same idea. As the cell gets too big, diffusion is it just doesn't work anymore. So we say transport becomes inefficient when the cell gets too big. So when the cell gets too big, what happens? Well, it divides. So how does the cell know when to divide? It's because of its size. As it gets too big, there are mechanisms along the way that tells the cell it's time to divide. So when we get to talking about efficiency, again, diffusion is only efficient at a very small size. Why cells cannot get very big? Because they rely on diffusion so much. That's one reason. And that's going to be done using a simple calculation called the surface area to volume ratio. And for that, you're going to watch a separate video at the website that shows you how to make those calculations, and then also gives you some examples and things you can work on your own. So this slide is not in your notes. You'll see some slides that come up that are not in your notes, but I just try to make your print-offs a little bit shorter. Otherwise it would have been 4 or 5 pages. So I'm going to move on from calculating surface area to volume ratio, which again just demonstrates why cells usually are pretty small, why they need to remain small. There's a certain ratio that you'll see. The larger the ratio, the better for the cells. So to relate that, we're going to do a diffusion lab with pink agar cubes that have a ingredient, chemical phenolphthalein. And just to kind of breeze over that lab really quick, not that you'll get many details here, we're going to save these two slides, this slide and the next slide, for the lab, but you'll see you'll get some cubes. Actually, you'll get 3 of these cubes, and they're going to sit in vinegar for about 10 minutes, and after those 10 minutes, that vinegar is going to diffuse in and basically eliminate some of that pink color where diffusion has taken place. And you're just going to chop them open, cut them basically in half, and you're going to see a measurement here, and that's going to give you a number that's going to help you calculate how much diffusion took place. Basically, the volume of diffusion is all the clear space where the pink does not exist. That's how far the vinegar went from outside in. But again, we'll get to these as we do our lab. Alright, so before we go any further, we're going to talk about cell cycle. I just wanted to show you a quick animation that is also located at my website, but just to kind of give you an idea of what we're talking about, I'm going to show it to you as an animation, and also ask you to draw this cycle on the back of your page that hopefully you're taking notes on right now. Alright, so here's that animation. Again, this can be found on my website. And your drawing on the back is basically going to be a circle with arrows. You can do that in just a minute, but here's where we're going to start talking about the cell cycle. The cell cycle comes in different phases. The first phase is called G1, and this is immediately after, which you'll see why it's a cycle. It's a circle. It just keeps going and going and going, so we got to start somewhere. This is just after the cell has divided. Okay, so naturally, just like anything that is brand new, it's going to be pretty small. The cell is going to start to grow. It's going to grow and develop, basically. That's what G1 is all about. The next phase is called the S phase, or synthesis phase. Synthesis is just a fancy word for making something, and in this case, it's making a copy of DNA. In order for the cell to divide, the DNA has to divide, and actually divide perfectly, otherwise the resulting daughter cells will either be non-functioning or malfunctioning, which could be devastating for a zygote, an early forming embryo. The next part is G2, and basically this G2 is preparing for cell division. It's growing a little bit more, but most importantly preparing for cell division. There are certain things that are necessary to get the cell ready to divide correctly. Again, if the cell does not divide correctly, the resulting 2 daughter cells could be devastating for that organism. Overall, G1, S, and G2 are referred to as interphase. You'll see that on the slide that you're on in your notes. Interphase consists of G1, S, and G2, and as you can see, interphase takes up most of the time of the cell life cycle. Basically about 3/4 of the cell life cycle, maybe a little bit more. Moving on from G2, we're going to go to mitosis, and we'll break down the 4 phases of mitosis a little bit later, but just so you know, mitosis is the nuclear division, so dividing the DNA. So first it makes copies of the DNA here in S phase, synthesis phase, and then during mitosis, that DNA is going to divide, so the 2 resulting cells again have equal amounts of DNA, otherwise it could be bad for that organism. And then immediately following mitosis is what we call cytokinesis, which is not described on here, but basically cytokinesis is cell division. Actually, a little more particularly, it would be called cytoplasmic division, because that's when the actual cytoplasm divides. Alright, so there's the cell cycle, a drawing that doesn't have to be 3D on your page, but just make a circle out of arrows, just like this, and label them pretty much how they are here, and anything else that I've added along the way if you want to. So pause the video, make this drawing. You might want to add cytokinesis. It's down here in the description. Cytokinesis is the C phase. We can add some details as we go, but make that drawing. Okay, you've got your drawing done, hopefully. Let's go back to our notes, flip back over to the front, and we're just going to describe the cell cycle as basically a period when the cell is simply growing and dividing, or you can just write when a cell grows and divides. That is the cell cycle. It's basically what the cell is doing. That's basically all of our cells, except for 2 exceptions. Just all they do is grow and divide. So hopefully you have that cell cycle diagram on the back, and again, if you make it big enough, you can add as much as you want to it. That'll be a good key to study from. But again, remember those first 3 arrows, G1, S, and G2, are all part of a big part of the cell cycle called interphase. An interphase basically is the cell carrying out normal cell functions. So what you can write here, carries out normal cell functions. It's doing whatever it's supposed to be doing. So go ahead and pause and write that down. Okay, G1, like I mentioned earlier, is just basically when the cell is brand new, so it's going to grow. Just like anything that's new, it's going to grow and develop. It's going to develop its organelles and other things. So grows and develops here in G1. And for S phase, again, remember S is synthesis. You may want to write synthesis here because that's going to help you maybe remember what's happening here. G1 and G2 just stand for gap. Gap 1 and gap 2, not as important. Synthesis, though, means to make something, and what it's making is a copy of DNA. So what you want to write here is synthesis of DNA. You can also add to that DNA replication, which again is another section of this next unit. So synthesis of DNA, otherwise known as DNA replication. And again, G2 comes after the S phase and right before mitosis, which is the beginning of the dividing part. So G2 basically is preparing for mitosis. So its location here right before mitosis kind of indicates what it's doing. It's preparing for cell division. It's preparing for nuclear division. So what you can write there, prepare for mitosis. And like I said earlier, there's also some growth taking place here, but most importantly, preparing for mitosis. So getting into the next one, mitosis. Just a simple definition. We're going to call this nuclear division. We'll get to all the steps and details later, but really quickly, nuclear division, or division of the DNA. We'll see how that works in those steps later. Last one, cytokinesis. And again, like I mentioned earlier, it's basically division of the cytoplasm. Cytokinesis is division of the cytoplasm. Alright, moving on to a slide that you don't have, but just to give you a quick idea, these chromosomes that you'll see during mitosis, and hopefully have seen these things before, have this basic X shape. But if you can see, zoomed in here, those X's are full of this tangled mess of DNA that has been wound up so tightly it's the only way to get 6 feet, approximately 6 feet of DNA into one single nucleus. One single cell in your body contains about 6 feet of DNA, give or take, depending on a few things, but approximately 6 feet of DNA, which is absolutely amazing that it can do this. And you can see here's the double helix, about the closest view you can have of DNA. And you can see then it goes down into just basically a string, which then gets wrapped around these proteins, which are called histones, we're not going to worry about. And then those things are also wrapped up and wound up. And so basically this thing is a very compacted mess that is very tightly packed in and organized, just full of DNA. So this is going to be the last slide. We're going to talk about the chromosome and how it relates to mitosis. You have a little bit different picture on yours that's a little bit easier to label, but this one's a little bit bigger and a little bit easier for me to go through with you. On a side note, we have 23 pairs of these guys, or a total of 46. Again, half from mom, half from dad, if you don't know where your DNA comes from. But we'll get into those details in a few units from now. What you're looking at here on the right is a double chromosome. Okay, it has two parts to it, basically two halves of the X. The long halves are the two halves. And this picture is a little deceiving. It shows a DNA in there, but this is DNA as it is not nearly as long as really what the chromosome is made of, like you just saw. Each half of the DNA, each one of these long halves, are called sister chromatids. That's one of your labels down here. And each sister chromatid is an exact replica. So again, during synthesis phase, when this DNA copies itself, this chromosome was once a single chromosome, copies into a double chromosome. So each sister chromatid is an exact copy of the other. And then in the middle here is a region of DNA where they actually are contacting each other. Each sister chromatid is touching each other. And that region is called the centromere right here. Centromere. And there's also some proteins that assist in that, but not a big deal. So again, mitosis is the nuclear division where this DNA is going to split in half. So why it makes this perfect X shape is so it can split nicely. If they don't split perfectly, this could be resulting in either mutations or cell death. I'm going to write those down. Either mutation or cell death.